Okay. So here's a really, really zoomed in micrograph of a myelinated nerve and an unmyelinated nerve. This one you see almost like an onion is wrapped in these sheets here. Numerous sheets created by that Schwann cell. In the center here is that axon, which is essentially just the inside of a neuron. So if you're inside an axon, you are inside that neuron. You're still part in that cytoplasm. It's just an extension of that. And then you see this unmyelinated axon here without those numerous myelin sheaths wrapping around it. So once again, the myelinated ones are the fastest ones. That's why if you're talking about like axons that go to muscles, if you look at the axons that reach out to muscle, like skeletal muscles, they're going to be much, much thicker and they're going to have a lot more myelin. Why would that be the case?
You need more voltage to get a response from other muscle fibers.
That's a good guess, but actually it takes the same amount of voltage to shock a muscle cell as it does like a gland in the body.
To get like a faster response.
Faster response. So you need to be able to respond. Your muscles are probably, they are the fastest responding tissues in your body, and they have to be in order for you to fight that bear, right? So if you're in a circumstance where you have to, it's life or death.
So we
Respond to external stimuli.
Oh, it's just one word. It won a bunch of awards.
That's just one word. What was that movie Leonardo DiCaprio just about a bear in?
He's in the wilderness.
Oh,
The Rebel.
Revenant.
Revenant.
Revenant. There we go. Yeah. Yeah. So if you are Leo DiCaprio in a movie on a movie set, you have to be able to
Fighting an animated bear.
Right, fighting an animated bear. You have to be able to do it fast, right? So, whereas the glands in your body are more slow to react, so those receive the unmyelinated nerve fibers. Definitely. Yeah, Sabrina and then Joe.
With the myelinated cells, maybe I am mistaking this for something else, but looking at the chapter, I believe it was chapter 4, we was talking about the structures of cell, like organelles. It
Chapter 5.
discussed chapter 5.
Or chapter 3 would be cells. Yeah.
Chapter 3, okay. It mentioned a disease caused by the mitochondria, the lysosome, and the peroxisomes. But the reason why I'm mentioning them is because it mentioned one, it was called Lenzo's oil.
Oh, Lorenzo's oil.
Lorenzo's oil. I want to know why would they mention the melanation of that associated with mitochondria or
Yeah, adrenoleukodystrophy is a disorder that Lorenzo had, and you may have seen this movie before, where it's an improper metabolism of the myelin. So eventually what happens is your myelin starts to degenerate. I can't remember if it's degenerating or building up. It's one of the two. I believe with MS it's degenerating. ALD it's building up. And so it clogs all the synapses. So if you can imagine having too much of that myelin, now you're suffocating the nerves, right? And so his cognition starts to decline. I watched that movie my freshman year in biology. I remember it's an interesting.
What do you call him?
Lorenzo's oil, and it's about that disease. And his dad's a scientist and he tries to come up with a cure while his son has this disease. It's really kind of an interesting movie. So yeah, too much myelin.
What does Russell Crowe have in that movie where he's a
Which movie?
John Nash.
Not the wrong movie.
The Beautiful Mind.
Beautiful Mind, there we go.
Oh yeah,
That's a story about John Nash. He's a brilliant mathematician and he has schizophrenia, but he's still a genius.
Yeah. Yeah, no myelin issues there. Just and actually it's
Just crazy.
We will talk about it. Yeah, he's crazy, right? He has hallucinations, but hallucinations have a foundation in science. I mean, there's a reason that they happen. So, yeah, go ahead.
I'm curious. I know you're going to talk about those other cells later, the
Neuroglia.
Is there a mechanism that keeps those myelin cells compositions in place? Like, if you were going to do the same routine with ions?
I guess I don't really understand the question.
Oh.
Rephrase it.
Because I'm thinking that there should be like an enzyme or response specifically to maintain the myelin. I keep saying all the other names, but I'm gonna say it. Myelin. Is there an enzyme that regulates the lipoprotein concentration, or make sure that it's not becoming too thick or too narrow?
Oh, I'm sure there is. I mean, there's an enzyme regulating everything. The name of it, I don't know what it is. I'll have to look it up and get back with you. But yeah, there would be a homeostatic mechanism of some sort of negative feedback that regulates an enzyme. Okay. Any other questions? Yeah, Joe.
Okay, so you're talking about myelinated versus unmyelinated axons. So in the nodes of Ranvier, they receive, another impulse that shoots it to the next node or whatever. So in an unmyelinated, is it just that first initial impulse that sends it down the axon and it doesn't receive any other impulses? That's why it travels so slow.
Yes, so it has to.
It doesn't have any other recharge points. So it.
Yeah, it's like if you can imagine like a really long wick, right? You just light part of the wick and you have to wait for it to burn all the way to the end.
Right.
Whereas on the myelinated axon, it would jump down the wick.
Okay.
Does that make sense? So it wouldn't take as long. Yeah. Cool. Does that answer your question?
Yeah.
Good. Okay. All right. So we have different types of nerves. So we talked about myelin, and then you have different nerves have different structures here in terms of lengths of dendrites and axons. Most nerves are multipolar. 99.9% of nerves are multipolar. Okay. And these, since 99% of nerves are in your central nervous system, that makes sense. All the multipolar neurons are in your central nervous system. Bipolar neurons are found in special senses, meaning we have one big long extension that branches out to a bunch of axons, and we have another big extension that branches out into axon terminals. So these are bipolar neurons. They're kind of equal. They've got bi means two, and then polar ends. And then the unipolar neuron has one extension, hence uni, and then that branches off into dendrites and axons. The sensory nerves are always going to be unipolar. Okay, so sensory neurons, those, especially the ones found in your skin, are going to be unipolar neurons. Those are the three main types. Okay, so the flow of information through the nervous system, before we look at what's happening molecularly at the cells. In general, you have a sensory receptor passing information into the central nervous system, which is divided here at this line. So you can consider this the spinal cord. That information is passed on into the gray matter of the spinal cord, which is the inside of the spinal cord. In there, it's going to either pass through an interneuron, which remember helps us communicate signals from sensory to motor, or sometimes it doesn't even bother with an interneuron, and that information just synapses directly onto a motor neuron. And then that motor neuron makes a decision whether to make some sort of change. So this is homeostasis here, just applied to the nervous system through these different neurons. Sometimes, excuse me, a decision is not made to send an impulse, okay? But you wouldn't know if it wasn't made, right? So most of the time, whenever you're doing something or something's happening, you're responding, that means we had this homeostatic feedback, this loop occur, okay? Great, an effector is going to be a muscle or a gland in general, something that carries out the effect of that neuron. And this is all an electrical event, guys, so this is all based upon electricity. We talked about the matrix. The human body does produce electricity, hopefully not for the machines. I hope we're not all hooked up to the fields, right? But we could be. Okay, neuroglia are going to do all these things. They're going to provide support for neurons. They're going to guide neurons into the right positions during embryological development, which just means your development as an embryo, as a fetus. Producing growth factors, removing excess ions and neurotransmitters you don't need, and actually even helping in forming synapses. So where two neurons come together, that is a synapse. And I asked you guys a question, I think the first week about synapses, and we're going to revisit that question today, is why do we have a space at the synapse? And we're going to get to that. I know everybody's just super excited about it, right? It's really pumped about it. Okay. Astrocytes are the first type of glial cell, and they are part of the blood-brain barrier. They look like this. So astro obviously referring to a star-shaped cell, okay? And it's got all these legs that reach out and cover blood vessels in the brain. So the astrocytes cover blood vessels, which is why they form the blood-brain barrier, or the BBB, right? And they are kind of like the Better Business Bureau because they regulate what substances are allowed in and out of the brain. Okay, they're a regulating agency, meaning if there is a bacterial cell here, or maybe a virus or a parasite, that won't be allowed through these tight junctions here. But if it's an ion or if it's a protein that the brain tissue needs, it's allowed through. So anything foreign is kept out of the brain by the astrocyte. Okay. And then oligodendrocytes. Yeah, go ahead.
Sorry.
So you're on.
When it comes to histamine and that blood-brain barrier, say if you're just taking the histamine to take care of an allergy.
An antihistamine?
Yeah, an antihistamine. How well does the astrocytes respond to that? Because I'm thinking, because some of the issues with antihistamines is that it's too effective in crossing the blood-brain barrier. And is that solely just due to the histamine itself, the antihistamine itself, or the additives with the antihistamine?
No, it's the shape of the molecule. So it's all chemistry. Whatever the shape of that molecule is will determine whether it gets into the brain or not. So it's almost like some molecules are trying to fit a square peg through a circular hole. That's kind of, the antihistamine medication just is the right shape to get through the barrier. So it all comes down to like, what's the chemical structure of that? Which is why it's so difficult to treat neurodegenerative disorders. A, we don't really understand them all that well, like their mechanism. B, when you try to come up with a drug, even if it would be an effective drug to treat that brain disorder, it may not cross that blood-brain barrier. So then the treatment's rendered useless.
So that's why it's so hard to treat MS.
Yeah, and well, MS is a problem because we don't really understand how it happens and we know what is happening to it.
Yeah, we don't know, but we don't know why the myelin continues to degenerate.
Right, yeah. We don't understand. MS, in case you guys are curious, you've probably heard of it. It's a disorder where the myelin, your body autoimmune attacks its own myelin, which doesn't make any sense. So that's why people are shaking their heads. It's like, why is this happening?
Autoimmune diseases don't make any sense.
No, they don't. It's a mutiny for no reason of your own immune system against your own tissues. So, yeah, we do know that it's genetically influenced, meaning some gene that gets switched on that causes your white blood cells to view the myelin as a foreign invader. Okay, and it's said to be triggered by another disease you got, where the molecules in that disease resemble the molecules in the myelin, so your body gets confused. And
It starts attacking the myelin instead of the actual.
Yeah, that's something that's actually dangerous. And so, yeah, this is weird. Yeah.
Just on a side note with that illness, there are like, because that one's predominantly female that are affected. But another interesting fact is that migraines also affect women more. So it seems like all of the diseases that affect women, like the doctors can't figure it out.
It's a conspiracy.
Yeah. Like heart disease, like all the symptoms for heart disease tend to apply to males.
Yeah.
But if it comes to females, they're like, oh, you have different symptoms. It's like,
yeah,
I think they need to know more about women anatomy and physiology. Even our textbooks show a lot of pictures of males.
Yeah, okay.
I don't know.
I'll have to go and count. Yeah, it just tells you how different the male and female body are, They experience disease differently, definitely. All right. I don't have anything for you there. So, okay. Oligodendrocytes myelinate the central nervous system. Microglia are just fancy white blood cells that only reside, they're kind of snooty, they only hang out in the coolest tissue in the brain and spinal cord, that's where they like to be, and they clean up stuff around there. And then ependymal cells are lining the ventricles of the brain, so they're responsible for producing and regulating the CSF.
The microglial cells, those are the little snowflake looking ones, right?
Those are, yeah, these guys here.
Right.
Yeah, it looks like snowflakes. They, I don't know, they're white blood cells. They're just trying to look like their friends. I don't know what, it's interesting, isn't it? It's like, you've got just the white blood cell, but if it's in the brain tissue, it's masquerading as a neuron. Maybe it's so it can sneak up on bacteria more easily. Okay, Schwann cells, we talked about those already. How they speed up impulse transmission through the axon. Satellite cells are the fifth type of cells seen in the peripheral nervous system in ganglia. Ganglia are just gatherings of cell bodies. Okay, it's just a cell body party in the peripheral nervous system. This is a ganglia. And you see them, actually a lot of times you see them just like in specific areas of the peripheral nervous system. So you see some ganglia or clusters of nerve cell bodies on top of really big arteries, because it's like a good area to like gather to then send out nerve signals to the surrounding important structures. And we talked about MS, it's a perfect segue into regeneration. So if you have, excuse me, MS mostly affects the peripheral nervous system. Okay, the spinal nerves and their attachments to muscles. If a peripheral axon is damaged, and it is in MS, it can regenerate, which is why MS gets better and worse and better and worse. It's your body regenerating, body degenerating, and it's a cycle of those two things. That's because the Schwann cells, okay, they will actually remain if the axon is damaged. So as long as you put both ends of the axon back together, okay, they will actually reattach and they will recover themselves in myelin. But in the central nervous system, oligodendrocytes don't remain after injury, which is the myelin-producing cell. So regeneration is unlikely because they no longer have that protective sheath. It won't recover. So even if you put those two axons back together, they won't wrap themselves up properly and send nerve signals again. So, that's why a stroke is much more difficult to recover from than tissue damage or a wound deep into the arm that severs nerves. They'll go in there, they'll find the ends of the nerves, they'll reattach them, they'll actually glue them together, and then they will start to send nerve signals again, which is pretty cool. So there are specific surgeons that do those surgeries. There's also surgeons that like mix and match nerves. So if you've got a neuron that's dead, they'll cut that nerve and reattach the loose end that goes to your fingers to another neuron in the surrounding area. I'm sorry, another nerve in the surrounding area to reestablish function to the fingers. It's really weird. It's kind of hard to wrap your brain around because, well, if this nerve was the nerve responsible for controlling your fingers, then how can attaching it to a spare nerve reestablish motor control of your fingers? But it does, because the brain is awesome.
It's like electricians.
Yeah, they're just going in and cutting wires and reattaching other ones. It's really neat. It's called a nerve transfer surgery. Yeah, Joe, and then
My dad kind of had the exact thing that you were talking about. He
Oh,
was severely
great.
burned when he was a child, and he was right-handed.
okay, not good.
And yeah, and the nerve, he was so severely burned that the nerves in his right arm, and he couldn't use his right arm until they like went in and repaired the nerves. And what was kind of interesting and cool, kind of, was that the scars that were left kind of looked like neurons, but he couldn't use his right arm anymore, so he had to learn how to do everything with his left.
Oh, okay.
Yeah. So, but eventually he regained the use of his right arm And
Oh.
he could do certain things, but he could no longer write or anything with his right arm, so he had to write left-handed.
Oh, so like fine motor control
Yeah.
skills were gone, but he could like flex his arm.
Oh
and
yeah,
extend
he
his
could
arm.
do just
Oh,
about
okay.
anything else once the nerves finally started to regenerate and everything
Yeah.
else. But yeah, he had to learn how to write with his left.
Yeah.
So
If you want to see how they do those surgeries, you just have to YouTube this. And they are really high quality videos, so you get to see all the muscles and stuff. Nerve transfer surgery, you'll probably enjoy it. It's really neat because they cut into the arm, and so you're actually getting to see the muscles, and she names them, and then she'll go through and she'll say, hey, this is the nerve I'm cutting, and then I'm reattaching this nerve. So you could learn a lot about the muscular system just by the patient's video. Huh?
I wonder what the patient's thinking.
Well, they're knocked out, so.
Who cares?
I'd be awake.
Right.
Just put a tourniquet on. It'll be okay. This is that regeneration process just shown here. So, you know, when we talked about the neuromuscular junction, where the neuron hits the muscle, this is that visual here. Now it's gone, okay? It can regrow through the Schwann cells that provide sort of like a guide or a map onto where it should reattach itself to the muscle, and it will reattach itself as long as it's not too big of a gap there, okay? So you need to put them close together. And that's what they'll do. In fact, a lot of the information we covered today will be reminiscent of what we covered in the muscular system, because the way that a nerve attaches to a muscle is the same way a nerve attaches to another nerve. So we're actually going to relearn the same thing, just apply it to another nerve, which is kind of nice. So before we do that, before we talk about the synapse, the neuron-to-neuron synapse, rather than a neuron-to-muscle, we have to talk about naming these neurons. The one reaching out and ending at the synapse is called the presynaptic neuron. And then the one receiving that signal, this guy here, is the postsynaptic neuron. And then in between the two is a synaptic cleft, or the space here. This connection between the pre and postsynaptic neuron is called the synapse. This is how that is visualized on a diagram. They will draw a little circle representing the cell body, an axon, and then a little Y ending. That's the presynaptic neuron. That Y ending will contact another cell body. We call that the synapse, and then it will send out its axon. So this is how it's depicted on a diagram, because you don't want to always draw out the whole nerve every time. We're talking about synapses. Okay, synaptic transmission is exactly the same as neuromuscular transmission. So what I want you to do is don't even look at the picture. Quiz your neighbor on neuromuscular transmission and see if they remember how to do it. If you're having trouble, okay, go back to that page in your notes. After you're done with neuromuscular transmission, I want you guys to do that first. Then we'll talk about synaptic transmission. Okay, ready to go. 4 steps.
Okay, there's some other info. What's the box?
that's the motor, that's the neuron.
Okay, alright, so in the motor neuron